Hello, my name is Eli Lyons, co-founder and CEO of Tupac.Bio. We provide software for synthetic biology research. And today I'm going to give a presentation on our method for demultiplexing sequencing reads for plasmid sequencing. So we actually developed this plasmid demultiplexing method due to the requirements of our own internal wet lab R&D work. And so in our case, we were transforming E. coli with multiple plasmids, and then we wanted to check the plasmid sequences at the end of the experiment. So the general problem we're solving is in the case that an experimenter has sequenced multiple plasmids from a single sample. So say you have multiple plasmids in an E. coli colony, or you have multiple plasmids in a mammalian cell transfection experiment, and you have reference sequences for the possible plasmids that are in the sample. But since you've sequenced these multiple plasmids together, you need to demultiplex. You need to determine which reads belong to which plasmids before doing more downstream analysis. And so we've resolved this problem with our demultiplexing workflow. So I'll give you some examples of where using our plasmid demultiplexing workflow may be useful. So one is to verify multi-plasmid transformation and transfection experiments are successful, or that those plasmids have persisted over time. Two, to identify copy number differences among plasmids in a sample. Or three, identify samples with mutant plasmid copies. So here's an overview of what we're trying to accomplish. And in this case, you're starting with mixed plasmid DNA from a single sample. So the assumption here is that you've purified your plasmid DNA. And in our case, in our proof of concept study, we have 3 different plasmids in that sample. And then you're performing sequencing. And for our case, in our method, we worked with nanopore sequencing and nanopore reads. So you have obtained these reads from sequencing, but you need to demultiplex them to figure out which reads belong to which plasmid. So we developed our demultiplexing method by using data that we collected that was related to our own wet lab R&D. And in this case, we have 3 plasmids, 2 of which have synthetic fragments in them. And as you'll see later, there's some similarity, especially in the backbone of these plasmids, which is part of the challenge for demultiplexing these sequencing reads. So what we found is that it's also very helpful to do data pre-processing to get a really accurate plasmid demultiplexing performed successfully. And so I'll go through our data pre-processing and plasmid demultiplexing algorithm. And again, in this case, we used 3 plasmids, and we started with plasmid reference sequences for all 3, which is important for our algorithm to work correctly. And the output after demultiplexing is reads that are binned into plasmid 1 reads and plasmid 2 reads, plasmid 3 reads, and unmapped or unclassified reads. And so after these reads are binned, you can do lots of downstream applications that we won't go into today. Here's our data preprocessing pipeline. And this pipeline is to be used in the case that you're either doing a run with DNA barcoded samples, or you have maybe done previous runs on the same flow cell with DNA barcoded samples, for example. So I think it's applicable to most cases. And of course, if it's not, you can just remove some of these steps. So we use Guppy for base calling and then remove the low quality junk reads, and then do demultiplexing with Guppy So Guppy demultiplexes the barcoded samples into bins. And then we take that data, and now we start to use our own steps. So in the BLAST demultiplexing crosscheck, what we're doing is we're looking for reads that have nanopore barcodes in the reads, and then removing those. And there is potential here for a lot of false positives if the reads have sequences that are correct, but very close to the DNA barcodes that nanopore uses. And so after that, then we run pore chop with the option discard middle, which is used to eliminate reads which have internal adapters or barcodes in the reads. So that's how we get rid of some reads that have potential to be demultiplexed incorrectly at later stages. So on this slide, we show how our data pre-processing can reduce incorrect binning of reads at the output. And so here you see the chimeric read percentage of the samples in our proof of concept study, as well as the switched barcode read percentage of the samples that we identified. Okay, so now we've gone through data pre-processing and we have filtered reads, and I briefly explained about the plasmids that were used in the proof of concept study that we have reference sequences for. And so now we'll move on to plasmid demultiplexing. So there's two major steps for our plasmid demultiplexing algorithm. The first step is to take the reference sequences for all of the plasmids that you expect to have reads for in the sequencing data. And then what we do is we use BLAST to find the similar regions from two or more plasmids, among two or more plasmids in those reference sequences. And so in this figure, those similar regions among the plasmids are shown in that dark red color. And these similar regions get deleted from the reference sequences for the demultiplexing purposes. You can use the full references for different purposes in the downstream analysis. But for our demultiplexing purposes, we delete these red similar regions. And then we classify reads by binning the reads to the unique regions in the modified reference sequences using BLAST. We performed a proof of concept experiment to verify our method and compare it with more naive or basic methods. And so the first step of this proof of concept study is that we took each of our three plasmids and we sequenced them without doing the transformation experiment where they're transformed together. So we took the plasmid DNA for plasmid 1, plasmid DNA for plasmid 2, plasmid DNA for plasmid 3, and we barcoded them separately, and then we performed sequence on a nanopore flow cell. And so in this case, we know the source of the reads for each plasmid, which would not be the case in the real experiment application that we would use this method for. But this allows us to check if we're correct in our demultiplexing or not, because we know where the original read is from. So after sequencing, we pooled the reads together in a single file. And this step mimics the real experimental setting that we would apply our demultiplexing method to. And of course, we still keep track of the source plasmid of each read. Then we run our method on this this file with pooled reads and classify the reads according to our method. And to assess the accuracy and sensitivity of our method, we compared our results with those obtained using other methods. And I'll go over those results next. When you have reads from multiple plasmids, the most naive way to demultiplex them would be to take each read and blast it against each of the plasmid reference sequences that are unmasked with the original reference sequence. So that's the most naive thing to do, and we did that just as a check to see what those results look like. And so in this table, you see that it's really difficult to get reads that match uniquely to a plasmid reference sequence. So in this case, for example, plasmid 1, you see most reads actually match to all three plasmid reference sequences by blast. And of course, you have a small number of unclassified reads. But this method is so naive and too poor in performance that we didn't even use it as a baseline. So instead of using the most naive method for our comparison, we made a baseline method that improves upon the naive method. So in our baseline method for comparison with our method, we take each read and we blast it against the original plasmid reference sequences, but we take reads that align to one and only one reference plasmid sequence as classified into that plasmid reference sequences bin. Okay, so we start binning the plasmid sequences if they only align to one reference uniquely. And in this case, we also included the chimeric reads and candidate switched barcode reads. They are not removed using our pre-processing algorithm. So in the baseline method, reads are classified uniquely to the plasmid references, but it's a very small number. And most of the reads are actually unclassified, and they never get binned or demultiplexed into the plasmid bins that would be useful for downstream analysis. So you end up with a lot of unclassified reads in the baseline method. However, in our method, you see that you have a lot more reads that are classified correctly and very, very, very few false positives. Looking at all of the reads, you can see that we classified 90% of the reads correctly, where the baseline method only classifies about 9%. So about a 10 times improvement. And our misclassified read percentage is virtually 0. Okay, so in conclusion, in this video, I went over our method, which is, as far as we're aware, a novel method to classify nanopore reads in the case that plasmids are sequenced together in a single sample. And in our proof of concept study, we show that our method compares favorably to a baseline method. And again, this method is practical for verifying multi-plasmid transformations and transfections or persistence of those plasmids in those experiments, or identifying copy number differences among the plasmids in a sample, or identifying samples with mutant plasmid copies. And this may be a useful demultiplexing method for other applications as well. And if there's interest, we may be providing this to our clients if they require it. For inquiries regarding this work or other bioinformatics work, you can send email to Eli, E-L-I, at Tupac.Bio. Thank you.